Swayam Prabha. Digital India. Educated India.
Welcome to Module 2 of the course Genome Editing and Engineering, where we are discussing about the breakage and repair of genomic DNA. In the last class, we discussed about the various methods by which or the phenomena by which the DNA of a cell gets broken. Today we are going to discuss how once this DNA is broken, the cell repairs it. So briefly we will be discussing today about the repair of genomic DNA. So this figure is now familiar to you. We spoke about the DNA damaging agents in the last lecture and how they creates different type of DNA damages. Now once the DNA damage has been inflicted, there are several DNA repair mechanisms inside the cell which takes care of the broken DNA, and if this DNA is left unrepaired, it will lead to mutagenesis and cause several genetic disorders. So what are the different types of DNA breakage repair systems inside the cell? So we have classified them into 8 main types, which may be photoreactivation or reversal of DNA damage, or base excision repair, nucleotide excision repair, mismatch repair, translation synthesis, interstrand crosslink repair or ICL repair, single stranded break repair, and double stranded break repair. In this lecture we will be discussing the first 7 repair mechanisms. The double stranded break repair will be discussed in detail separately. Let us start with the first type of repair that is used by the cell whenever a DNA is broken. This is the photoreactivation or reversal of DNA damage. So what is photoreactivation? Photoreactivation. It is a process by which UV inactivated organisms restore their function by repairing UV induced damages within the energy of near UV light, which is 310 to 410 nanometers, and this is done with the help of an enzyme called photolyase. UV photolesions and alkylated bases are 2 types of DNA lesions that can be easily reversed in an error-free way in lower organisms and mammals through photolyase mediated photoreactivation of UV lesions. So once the DNA is damaged by UV light, it will create certain lesions, and these lesions are repaired with the help of these enzyme. Now what are the different kinds of UV induced damage? So depending on the type of the UV, so you have UVA induced DNA damage, as you can see in the left arm marked by the number 1, or you may have UVB induced DNA damage in the right arm represented by the number 2. So once UVA induced damage occurs, it leads to oxidative stress and DNA photoproducts like 8-oxo-deguanylations are formed. UVA induced damage may also lead to immunosuppression in certain cases, and whenever there is a DNA photoproducts as a outcome of this damage, it leads to the mutagenesis. Similarly, due to UVB induced damage, certain DNA photoproducts are produced like cyclobutene pyrimidine dimer or 6,4-PP, and similar to the UVA induced DNA photoproducts, they also lead to mutagenesis inside the cell. And when there is mutagenesis due to any of these UV induced damages, it leads to failure of apoptosis, and which may lead to some kind of cancers. We will discuss this in detail in a stepwise manner. So, let us first discuss the CPD or cyclobutene pyrimidine dimer repair via photolyse activity. So, as you can see that whenever a UV ray is hitting the DNA, there is a formation of cyclobutene pyrimidine dimer or CPD dimer in the DNA. Now, in the presence of visible light, the photolyse binds to this CPD lesion and repairs it, and thereby reverting it back to the monomeric DNA. What is the nature, structure and function of this particular enzyme which carries out this kind of reversal reaction in the presence of visible light? So, DNA photolyse is basically a monomeric flavin dependent repair enzyme with 420 to 616 amino acid residues, varies from species to species. It contains two known cofactors, a catalytic cofactor and a light harvesting cofactor. The reduced flavin adenine dinucleotide FADH minus work as the catalytic cofactor in all photolyses. It transfers energy to the cyclobutene pyrimidine dimers in the form of an electron, breaking the cyclobutene ring and producing two monomeric bases. And you can see the location of the reduced FADH over here. This is the ribbon structure of photolyse, DNA photolyse, and here you can see the surface potential representation. There is a dashed box over here which marks the hole leading to FAD. The positive blue colors and the negative red colors charged residues are represented in this figure in a color code. Now, going back to the structure of these enzyme, the light harvesting cofactor include 5-10 MTHF, 8 hydroxy 5-deazoriboflavin or 8 HDF and FMN. They absorb light energy and transfer them to FADH reduced. Let us see the photoreactivation reaction a little bit of more detail. Whenever there is UV exposure, this causes pyrimidine lesion, for example, the thymidine dimer formation, which we have discussed now and then. Whenever these thymine dimer formation takes place, the photolyse or the photoreactivating enzyme detects these damage. The antenna molecule of the photolyse, such as the MTHF, 8-HDF and FMN, here you can see them, trap the light energy and transfer it to the catalytic cofactor FADH, which becomes excited, and it transfers energy to the pyrimidine dimer in the form of an electron, thereby splitting the CPD into two monomeric units, and then the electron is transferred back to the flavin molecule, and in this way the damage is repaired. What happens in humans and other mammals? In these higher organisms, two different classes of enzymes reverse the alkylated bases. These are O6 alkyl guanine DNA alkyl transferase or AGT, MGMT, then O alkylated DNA lesions are reversed with this AGT, MGMT, and like O6 methyl, ethyl, 2-chloroethyl benzene and aliphatic groups. Let us now discuss how similar lesions are repaired in humans and other mammals. In them, there are two different classes of enzymes which reverses the alkylated bases. In the first case, O6 alkyl guanine DNA alkyl transferase enzyme can reverse O alkylated DNA lesions like O6 methyl, ethyl, 2-chloroethyl, benzene and aliphatic groups, the guanine pyridoyl oxybutyl adducts or the GPD adducts. It can also repair the O6Z alkyl O6Z intrastrand crosslinks. A single AGT molecule can eliminate the alkylation adduct in a single step reaction by transferring the alkyl group from the oxygen of the DNA base to the cysteine residue in its catalytic pocket. In a second case, the ALK-B related alpha ketoglutarate dependent dioxygenases ALK-B mediates reversal and it reverses the N-alkylated base adducts. ALK-B family proteins hydroxylate an alkyl group in a alpha ketoglutarate and iron 2 dependent manner for demethylation. The oxidized alkyl group is released as formaldehyde, leaving behind the original base. Let us now go to another type of DNA damage repair, which is known as the base excision repair or BER or BER. The oxidative deamination, alkylation and abasic single base damage types that do not significantly deform the DNA helix are corrected by base excision repair. Base excision repair is mostly active in the G1 phase of the cell cycle. Base excision repair mechanism requires chromatin remodeling at the site of DNA damage, followed by detection of the DNA lesion by DNA glycosylase. Many different glycosylases are involved in base excision repair, each of which distinguishes and eliminates a specific modified base or bases from the DNA. These DNA glycosylase can be classified into two categories in terms of their function. The first are the monofunctional and the second are the bifunctional. The monofunctional with only glycosylase activity, example uracil glycosylase, mu2I homolog and N-methylpurine DNA glycosylase. Bifunctional have both glycosylase and beta lyase activity, and examples are DNA glycosylase 1, Ni-like DNA glycosylase 1 and Ni-like DNA glycosylase 2. In addition, a few DNA glycosylases can act as both mono and bifunctional enzymes. Examples include Ni-like DNA glycosylase 3 and 8-oxoguanine DNA glycosylase. Monofunctional and bifunctional DNA glycosylases vary in their DNA damage source pattern, recognition of the damage site and excision. Monofunctional uracil and glycosylase or UNG searches for uracil in DNA through random interaction, forming an open conformation. Once it comes in contact with DNA, it transforms to the closed conformation for base interrogation. The selected bases are then flipped out of the DNA duplex and put into the catalytic pocket of the enzyme, where specific hydrogen bonds align the base for nucleophilic attack by an activated water molecule that has been placed by a conserved aspartic acid residue. It then removes the recognized base through the catalytic cleavage of the glycosidic bond, leaving an abasic site without disturbing the phosphate sugar DNA backbone. The abasic site is subsequently repaired by a series of enzymes that cleave the backbone, insert the replacement residue and ligate the DNA strand. So, many functions being carried out at the same time. The A basic sites produced by monofunctional DNA glycosylases are repaired through short patch repair pathway. On the other hand, bifunctional enzymes initiate the long patch repair pathway after the formation of A basic sites. In short patch repair, AP endonuclease act on the A basic site, which breaks the phosphodiester bond 5' to the A basic site and produces a hydroxyl residue at the 3' end, leaving a deoxyribose phosphate at the 5' end. Tailoring of this repair gap is carried out by the 5' DRP lyase activity of Pol beta, followed by filling the single nucleotide gap by Pol beta and ligation by either DNA ligase 1, Lig1 or a complex of DNA ligase 3, Lig3 and XRCC1, which is X-ray repair cross complementing protein 1. In the long patch repair pathway, the repair gap tailored by the 3' phosphodiesterase activity of AP endonuclease 1. Thereafter, Pol B or Pol delta epsilon fills the gap, particularly in proliferating cells, in a strand displacement manner, followed by flap removal by the flap endonuclease and a DNA ligase 1 mediated ligation. So, this figure shows the mechanism of base excision repair pathway. In figure 1 on the left side, you can see damage specific DNA glycosylase recognize and remove the damaged DNA base, resulting in an A basic site. So, this is the DNA glycosylase. So, APE1 binds to this A basic site and cleave the DNA backbone, and you can see here the outcome. This results in a single strand break with a 5' DRP or 5' deoxyribose phosphate moiety. The DRP lyase activity of Pol beta removes the 5' DRP and inserts a new undamaged nucleotide into the gap. Finally, the Lig3A XRCC1 complex completes short patch base excision repair by sealing the remaining nick in the phosphodiester backbone. So, here you can see the damage, and finally the damage is being healed at the end of this pathway. Let us go to the second case, where if the 5' DRP moiety is resistant to the DRP lyase activity of Pol B, a polymerase switch to Pol delta epsilon takes place. So, here this Pol B is ineffective. That is the reason why these switched to this side of this reaction arm, where Pol delta and epsilon will be helping it in repairing the damage. This generates a 5' flap structure, which is recognized and excised by FAN1 or flap endonuclease 1. So, this will be removed by this FAN1 in a proliferating cell nuclear antigen dependent manner. To complete the patch base excision repair, DNA ligase 1, here it is Lig3, here it is ligase 1, in association with the PCNA seals the remaining nick in a DNA backbone, and this is getting resolved or repaired through the long patch base excision repair. Let us now go to the third type of DNA damage repair, which is called as the nucleotide excision repair or NER. Bulky DNA lesions such as CPDs or cyclobutane pyrimidine dimers and 6,4 photoproducts, 6,4 PPs resulting out of UV radiation, benzoapyrene adducts and some other DNA damages are repaired by the nucleotide excision repair mechanisms. The two major branches of NER, one is the global genome NER, another one is the transcription coupled NER. Briefly they are known as GGNER and TCNER. We will discuss their mechanisms one by one. GGNER can occur anywhere in the genome, whereas TCNER is responsible for the accelerated repair of lesions in the transcribed strand of active genes. So, you can see here the transcription coupled repair in the left arm, and you can see the global excision repair in the right arm. And the players involved in the two process differ from one another in the starting. Towards the end, they merged to the same pathway. So, let us see this figure keenly. The DNA damage that is happening over here is recognized differently depending on whether DNA is transcriptionally active or transcriptionally inactive. If the DNA is transcriptionally active, transcription coupled repair will take place. If it is inactive, the global excision repair will take place. After the initial recognition step, the damage is repaired in a similar manner as already told to you, with the final outcome being the restoration of the normal nucleotide sequence. So, here you can see the damage in one of the strands, and finally, this damage is taken care of and the normal nucleotide sequence is restored. So, we will not go into details of the various factors and enzymes which are involved in this mechanism, those are shown here in this figure. For your interest, you may study these pathway to understand the mechanism in detail. As already told to you, the global genomic nucleotide excision repair can occur anywhere in the genome, whereas TCNER is responsible for the accelerated repair of lesions in the transcribed strand of active genes. GGNER is initiated by the GGNER specific factors XPC RAD 23B, in some cases with the help of UV-DDB or UV damaged DNA binding protein. In GGNER, two protein complexes are associated with the detection of DNA damage. The first protein complex is composed of XPC, Geroderma pigmentosum group C, RAD23B, UV excision repair protein RAD23 homolog B, and centrin 2, a protein encoded by the CTN2 gene. At first, this complex recognizes the DNA damage that disrupts the normal Watson-Crick base pairing. The XPC protein contains two hairpin structures. In the second stage, the DNA binding domain of XPC binds with the non-hydrogen bonded bases of double stranded DNA and inserts a beta hairpin through a DNA duplex. This causes the damaged base to flip out of the DNA double helix. The second complex is UV binding DNA damage protein consisting of DDB2, DDB1, CAL4A or CAL4B and RBX1. This complex enhances the binding of XPC, RAD23B and centrin 2 protein complex to the damaged DNA. Global genomic nucleotide excision repair involves the UV-DDB complex, which generally binds with UV generated lesions, including pyrimidine, pyrimidone photodimers or 6,4-PPDs and cyclobutane pyrimidine dimers or CPDs. It also recognizes DNA with epurinic or apyrimidinic or epi sites and base misbases. NER excises 24 to 32 nucleotide DNA segments containing a broken link with extreme precision in higher eukaryotic cells. The final stages of DNA repair is reparative synthesis, utilizing an undamaged strand as a template, followed by ligation of the single strand break caused by the damage. So, this is the schematic representation of GG NER pathway. Wherever a DNA damage occurs, as already discussed to you, XPC-HR23B detects it, and this follows a cyclic pathway, and at the end of which the DNA is repaired and the final nick sealing is done with the help of ligase 1 or ligase 3. There are various proteins involved in this pathway, the details of which are listed in this table, and these proteins have carry out different functions. For example, this XPC-HR23B is used for the recognition of the distorted DNA structure, as already told to you. Then DDB recognizes damage and it interacts with the chromatin. XPA has a structural function binding to a damaged strand. RPA, it binds to a single stranded DNA. Then TF2H, it has ATPase activity, minor helicase activity, 3' to 5' DNA helicase activity. XPase have endonuclease and catalyzes the formation of single stranded break in DNA on the 5' side of the damage. XPZ, it is an endonuclease which catalyzes the formation of single strand break in DNA on the 3' side of the damage. PCNA is a factor which ensures processivity of the DNA polymerases. Pol delta, Pol epsilon, they are DNA polymerases. Ligase 3 and ligase 1 participates in the ligation of the single strand breaks, and RFC is a ATP dependent connection of the PCNA. Let us now examine the transcription coupled nucleotide excision repair or the TCNER. This is a specialized nucleotide excision repair system known as transcription coupled nucleotide excision repair, which repairs DNA damage in the transcribed strands of active genes. The TCNER pathway is initiated by helix distorting lesions that block the progression of elongating RNA polymerase II. The halted RNA2 complex triggers the recruitment of ERCC6 or excision repair 6. ERCC gene encodes a protein called cocaine syndrome B, CSB. The ERCC6 protein recruits ERCC8 or CSA protein. ERCC8 is a part of ubiquitin ligase complex. The ubiquitin ligase complex also includes DDB1, Cal4A or Cal4B and RBX1. The ERCC8 ubiquitin ligase complex is one of the key regulators of TCNER, which causes ubiquitination of one of the more factors involved in this repair process, including the blocked RNA Pol2, ERCC6 and transcription elongation factor TF2H. The TCNER pre-incision complex also contains XPA, XAB2 complex, ACE-MGN1, TCAN1, UVSSA in complex with USP7 and EP300. The probable role of XPA is to help in the assembly and stability of the pre-incision complex. XBA2 is required for pre-mRNA splicing and also modulate the structure of the nascent mRNA DNA hybrid through its RNA DNA helicase activity. TCEA1 helps in the backtracking of the RNA pol2. This allows the repair proteins to get access to the damaged site. So, this figure is a model of CSB's distinct activities in transcription coupled DNA repair. So, you can see here on the top the targeting of CSB to lessen stalled transcription. So, due to this lesion here, as already discussed earlier, the RNA polymerase II cannot progress further and therefore, it is stalled. CSB has two distinct actions once recruited. In this step, you can see the recruitment of the CSB. Protein factor recruitment is one action that occurs independently of nucleosome repositioning. Components of the nucleotide excision repair complex, the CSA-CSN, E3 ligase complex and the TF2H transcription elongation complex are all recruited at this stage. CSB also recruits NAP1, L1 and NAP1, L4 to lesion stalled transcription sites. In the lower right, we can see the nucleosome repositioning, which is required for a second action. CSB moves nucleosomes in combination with NAP1 like histone chaperones to allow effective DNA repair and or continuation of transcription following the repair. Let us move to the next type of repair, which is known as the mismatch repair. The mismatch repair mechanism fixes short insertion and deletion loops within repetitive DNA sequences that have resulted from strand slippage events and base mismatches produced during the DNA replication. Mismatch repair pathway is highly conserved whether in eukaryotes or prokaryotes and mainly associated with DNA replication, increasing replication fidelity by around 100 folds. Mismatch repair consists of the following basic steps. In the first step, a detector or sensor marked as homolog is employed to detect or identify the mismatch. The sensor then activates a set of proteins, a MAT-L homolog and an exonuclease that select the nascent DNA strand to be repaired, nick the strand, exonucleotically remove a region of the nucleotides containing the mismatch, a DNA polymerase resynthesizes the strand and a DNA ligase finally seals the remaining nick. Let us study the E. coli DNA mismatch repair pathway. The following proteins are required for E. coli MMR pathway: MAT-S, MAT-L or mutator S, mutator L, MAT-H and finally DNA helicase 2. What does MAT-S do? As already told, it can recognize base mismatches and small nucleotide insertion or deletion. MAT-L interacts directly with the MAT-S and enhances its ability to recognize mismatches, recruits and activates MAT-H. MAT-H can specifically incise the unmethylated nascent DNA strand of hemimethylated DGATC, which acts as the initiation site for the mismatch provoked excision. DNA helicase 2 generates single stranded DNA once it is loaded at the nick and opens the duplex from the nick towards the mismatch. The single stranded DNA binding protein SSB is then rapidly bound by single stranded binding protein to prevent it from duplex formation. Let us now discuss the E. coli DNA mismatch repair pathway in a little bit of details. The various proteins required for this MMR pathway in E. coli like MAT-S, MAT-L, MAT-H, then DNA helicase 2 endonucleases, and DNA polymerase III holoenzyme, and finally the DNA ligase. So, these proteins carry out various functions in this repair pathway. For example, MAT-S recognizes the base mismatches and small nucleotide insertion deletions. The MAT-L interacts directly with MAT-S and enhances its ability to recognize these mismatches, recruits and activates the MAT-H. The MAT-H can specifically incise the unmethylated nascent DNA strand of hemimethylated DGATC, which acts as the initiation site for mismatch provoked excision. The DNA helicase 2 generates single-stranded DNA once it is loaded at the nick and opens the duplex from the nick towards the mismatch. The single-stranded DNA binding protein SSB is then rapidly bound by single-stranded binding proteins to prevent from duplex formation. In the fifth step, the exonucleases, which are XO1 or XOX and have 3' to 5' exonuclease activity, or XO7 or RECJ, which has 5' to 3' exonuclease activity, excises the nicked strand from the nicked site up to and slightly past the mismatch depending on the location of the strand break relative to the mismatch. DNA polymerase III holoenzyme resynthesizes at the single-stranded gap while the DNA ligase seals the nick. In human and other eukaryotes, the MMR process is almost similar and controlled by homologs of the above proteins as discussed. So, in this figure, we can see the eukaryotic MMR, the MAT-S alpha, which recognizes base-base mismatches, and MAT-L alpha, it nicks the 3' to 5' side of the mismatched base of the discontinuous strand in figure A. In figure B, we can see the MMR in MAT-H-less bacteria. Mismatched bases are recognized by MAT-S and incision of the discontinuous strand by MAT-L. In figure C, we can see the E. coli MMR MAT-S recognizes mismatched bases and MAT-L interacts with and stabilizes the complex. Then MAT-H endonuclease is activated to incise the unmethylated GATC site to create an entry point for the excision reaction. Let us now examine the reaction in eukaryotes as well as in E. coli. So, you can see in figure A the eukaryotic MMR where MAT-S alpha recognizes base-base mismatches and MAT-L alpha nicks the 3' or 5' side of the mismatched base on the discontinuous strand. In B, you can see the MMR in MAT-H-less bacteria where mismatched bases are recognized by MAT-S and incision of discontinuous strand by MAT-L. In figure C is the E. coli MMR. Here MAT-S recognizes mismatched bases and MAT-L interacts with and stabilizes the complex. Then MAT-H endonuclease is activated to incise the unmethylated GATC site to create an entry point for the excision reaction. In figure A, you can see the resulting DNA segment is excised by XO1 nuclease in cooperation with the single-stranded DNA binding protein RPA. In the figure B, you can see the error containing DNA strand is removed by the cooperative function of DNA helicases such as UVRD, the exonuclease RECJ and XO1, and the single-stranded DNA binding protein SSB. In the case of E. coli MMR, the DNA helicase, a single-stranded DNA binding protein, and several exonucleases are involved in the excision reaction. In eukaryotes, as seen in figure A, the DNA strand is resynthesized by DNA polymerase delta and DNA ligase 1 will do the ligation reaction, whereas in MUD-H-less bacteria, DNA polymerase III and DNA ligase does the job. In E. coli, the DNA polymerase III and the DNA ligase fill up the gap to complete the repair. So, this is a comparison between the eukaryotic MMR and E. coli MMR and with MMR in MUD-H-less bacteria. Let us now go to the fifth type of repair mechanism, which is the translation synthesis. Cells copy DNA that has unrepaired damage that blocks the replication fork from moving further through a process called translation DNA synthesis, or TLS. It is carried out by highly conserved TLS polymerases. There are a total of 11 TLS polymerases reported, and these are divided into Y, B, X and A families. This damage tolerance mechanism is frequently involved in incorporating erroneous bases, and the cells survive as a outcome of these, but are often associated with an increased risk of carcinogenesis and mutagenesis. Let us discuss the polymerase switch model of translation synthesis. The other model is the gap filling model. In polymerase switch model, the TLS polymerase combines sequentially in a two-step procedure to replicate through the DNA lesion at a forked replication that has halted. A nucleotide opposing the DNA lesion is first incorporated by an inserter TLS enzyme, which is typically a Pol L and Pol K, and less frequently Rev1 or Pol zeta. So, there are two types of translational synthesis. One is the polymerase switch translational synthesis, or the polymerase switch model. The other one is the gap filling model. In the polymerase switch model of translational synthesis, there are two sequential steps to replicate through the DNA lesion at the forked replication, which has been halted. A nucleotide opposing the DNA lesion is first incorporated by an inserter TLS enzyme, and it has various members, as you can see in this list. In the second step, the primer template termini are extended by an extender TLS enzyme, which typically do not always is performed, but in some cases by another enzyme called Pol K. In the gap filling model of translation synthesis, TLS synthesis machinery targets the single stranded gaps left behind during replication by replicative polymerases or through an incomplete DNA repair process. The precise sequence of events for a gap TLS is still unknown. However, experiments on mouse cells have shown that Rev1 plays a crucial role in post-replicative gap filling. Similarly, Rev3 is important for TLS across gaps opposite to 6,4 photoproducts. Both Rev1 and Rev3 are error-prone DNA polymerases which function as inserter and extender polymerases. Let us now discuss the sixth type of DNA repair, which is the interstrand crosslink repair. Interstrand crosslinks are DNA lessons in which two bases from complementary strands are covalently linked due to the activity of various cross-linking agents such as nitrogen mustards, mutamycin C, alkylating agents, etc. They are highly toxic DNA lessons and can block DNA from transcription and replication by inhibiting DNA strand separation. The Fanconi anemia proteins identify and treat these lesions. Interstrand crosslink repair is initiated in a cell cycle dependent way by chromatin loading of the FA proteins. Let us discuss the ICL removal in G0 G1 phase cycle. In G0 G1 phase of cell cycle, ICL removes as described below. Recognition of an ICL by NER machinery takes place. In ICL block transcription, CSA and CSB, two specific factors for transcription couple NER are needed to load the incision complex. On the other hand, for ICLs located in non-transcript regions, the XPC HHR23B damage recognition factor complex loads the NER incision complex. The first incision is introduced by the incision complex composed of XPA, RPA, TF2H and XPG. The ICL lesion with the oligonucleotides is bypassed by TLS polymerases after the first incision. The second incision is introduced by another NER incision complex. So, here we can see the ICL repair model in quiescent cells or G0 G1 cells phase. The NER machinery recognizes an ICL on the DNA. In the event of ICL blocked transcription, two transcription couple NER specific factors CSA and CSB are required to load the incision complex. In contrast, for ICLs in the non-transcribed areas, the XPC HHR23B complex is in charge of loading the NER incision complex. The incision complex comprising XPA, RPA, TF2H, XPF, ERCC1, XPG initiates the initial incision. Following the initial incision, a TLS polymerase such as DNA polymerase or REV1 bypasses the ICL lesion with the oligonucleotide. Another NER incision complex initiates the second incision. ICL repair in S phase. ICL lesions can block DNA replication forks. The FANCM FAP24MH F complex binds to a stall replication fork and recruits both the FA core complex and BLA top 3 alpha RM1 BTR complex. Active FA core complex monoubiquitinates both FAN-CD2 and FAN-C1, which allows incisions of the ICL using unique structure specific endonucleases such as XPF, FAN-CQ, ERCC1, SLX4, FAN-CP, SLX1, MAS81, EME1 and FAN1. The incision initiates a double strand break which is repaired through homologous recombination. Both RAD51 recombinase paralogs and BRCA complexes are required for the formation of RAD51 filaments at damaged sites. These double strand breaks mechanisms will be discussed in detail separately. Let us now go to the seventh repair mechanism, the single strand break repair. These single strand breaks are caused by oxidative damage to the DNA, abasic sites, errors made by DNA topomerase 1, etc. Unrepaired SSBs cause DNA replication stress and transcription pausing and overactivation of PARP, resulting in genomic instability and human illnesses such as cancer, heart failure and neurological disorders. These SSBs are repaired by various DNA repair mechanisms. There is a canonical SSB repair pathway which is the rapid global SSB repair mechanism and includes SSB detection, DNA end processing, DNA gap filling and DNA ligation. Sometimes the SSB repair pathway is considered as a specialized sub-pathway of base excision repair as discussed earlier. PARP1, poly-ADP ribose polymerase 1 and X-ray repair cross complementing protein 1 or XRCC1 play key roles in the rapid global SSB repair pathway. Recent studies have also showed that SSBs can also be repaired by either homologous recombination or alternative homologous mediated SSB repair pathway. Depending on the source of the SSB, SSB repair occurs by one of the three possible mechanisms. The first one is in the long patch pathway. SSB are transiently identified by poly ADP ribose polymerase 1 or PARP1, which undergoes a rapid cycle of poly ADP ribosylation before dissociating to detect the subsequent SSB. Then the ends are processed by the epiremidic endonuclease 1 APE1, PNKP and APTX. After that, the damaged 5' termini are removed by FAN1 in association with PARP1 and PCNA. The gap is then filled up by polymerase beta in association with delta and epsilon. Finally, the ligation is performed by Lig1 with the help of PCNA and XPCC1. In the short patch pathway, AP1 recognizes SSBs produced during base excision repair, which then uses a end processing pathway similar to the long patch repair. However, only the polymerase beta enzyme performs the gap filling step, which is followed by ligation catalyzed by Lig3. In top 1 SSB pathway, the end processing is carried out by the TDP1, which eliminates top 1 from the 3' end. Double strand breaks generated from SSBs during DNA replication can cause chromosomal breaks and translocations, resulting in severe genomic instability if not repaired quickly, and these are addressed by the double strand break pathways. So, you can see here the left arm is having endogenous reactive oxygen species and hydrogen peroxide. Basically, these are oxidative assaults by these reactive oxygen species, and in the right arm or pathway 2, aberrant top 1 activity. PARP1 is one of the first proteins to be attracted to the SSB site. Poly ADP ribose glycohydrolase also regulates poly ADP ribosylation or PARG. Following the damage detection step, X-ray repair cross complementing protein 1, XRCC1 recruits several key proteins required for broken DNA processing, DNA end processing and DNA ligation to the sites of damage, including DNA polymerase, polynucleotide kinase, APTX, tyrosyl DNA, phosphodiesterase 1 and DNA ligase 3. This gap filling requires either DNA ligase 1 or Lig3 depending on the size of the gap. End resection is important in SSB repair. The enzymatic end processing at SSB site may be performed in two directions, 3' to 5' or 5' to 3', which are referred to as 3' 5' SSB resection and 5' 3' SSB end resection respectively. In cell-free egg extracts of genopas, oxidative DNA damage derived indirect SSBs are reported to be processed by AP2 in the 3' to 5' direction to promote ATR-CHQ1 DNA damage response pathway, which stabilizes replication forks and prevents generation of DNA double strand breaks. Few enzymes associated with DNA metabolism such as TDP2 and APTX can digest SSB ends in the 5' to 3' direction, suggesting a possible mechanism of 5' to 3' SSB end resection. So, in this figure, 4 steps of 3' to 5' SSB end resection is being shown. In the first step, SSB end sensing and processing takes place. In the second step, initiation of the SSB end resection takes place. In the third step, continuation of the SSB end resection happens, and there is finally a termination of the end resection at one particular point, and as a result of these a SSB DNA gap is created. What happens if there are defects in DNA repair and all the 7 different DNA repair mechanisms fail to operate? This will lead to various kinds of genetic diseases, as told in the beginning of these lectures. Some of the diseases that may occur due to defects in the DNA repair mechanism are Fanconi anemia, xeroderma pigmentosum, cocaine syndrome, trichothiodystrophy, and there are many. These are some of the diseases, and the list is endless. As already told to you in the beginning of the lecture, if the DNA repair mechanisms fails, it may lead to many mutations and genetic diseases. The cell is quite rich in its repertoire of DNA repair mechanisms, and we have discussed in detail about 7 different DNA damage repair mechanisms, and we are going to discuss about the double stranded break repair in detail separately. These are some of the examples of genetic diseases which may occur due to failure in the DNA repair mechanisms. For example, Fanconi anemia, xeroderma pigmentosum, cocaine syndrome, trichothiodystrophy, etc. But there are many other which you can find out as a small assignment. Thank you for your patient hearing.